special components called organelles. There are three main components for all the cells. They are cellular membrane, nucleus and cytoplasm. Today we are going to learn about plasma membrane. It is the outermost covering of the cell. It separates the cell components from its external environment. And it is very flexible and it is made up of protein and lipid. Plasma membrane has another name, that is selectively permeable membrane. That means permeable, permits or allow. It allows only some substance to entry and exit of the cell. That's why it got the names selectively permeable membrane. Now let's see, how does the movement of substances taking place inside and outside of the cell? Different processes are helping for that. First one is diffusion. I'll tell you some examples for diffusion. If you spray perfume in your body, only you people will get the smell? No, the smell will be spread everywhere. That is one example of diffusion. That means that smell particles are spread around you. Another example I will tell you. You are sitting inside your bedroom. Your mother is making biryani inside the kitchen. You will get the smell. You will run to the kitchen, isn't it? So that means the smell particles are spreaded there or diffuses there. One more example I will tell you. If you keep a tea bag in a glass of hot water, the water will become tea because that tea powder was diffused in the water. So these are the examples of diffusion. So what is diffusion? Diffusion is the movement of substances from higher concentration to a lower concentration. You know, carbon dioxide is a cellular waste. So if it is high inside the cell, it will be moved from the cell to the outside of the cell. Like that, you know, cell need oxygen for all the activities. So when the oxygen content is less inside the cell, the oxygen will be enter into the cell from the outer environment. So like this only, carbon dioxide and oxygen is moved inside and outside of the cell by the process of diffusion. This solution taking place through a semi-permeable membrane. It is known as osmosis. That means osmosis is the movement of water from the higher concentration to a lower concentration. So you know our plasma membrane is selectively permeable. So the water movement through the semi-permeable membrane from the higher concentration to the lower concentration by osmosis. Now you understand what is diffusion and what is osmosis and how the movement of carbon dioxide and oxygen and water taking place inside the cell. Some other substances are there. Their movement taking place inside the cell by using energy. Children, now we are going to learn about different kinds of solutions. Before that, you have to know what is solution. Solution is a mixture of solute dissolved in the solvent. Now I am going to draw three beakers here. Inside this beaker, I am going to keep a cell. Then each beaker containing sodium chloride solution. In that sodium chloride solution, 50% is sodium chloride. You know it is salt. That means 50% solvent, that is water. So all the beakers contain the same amount of sodium chloride solution. That means 50% sodium chloride and 50% water. Then in the first beaker, inside the cell having 20% sodium chloride. That means 80% water. Here, inside the cell having 50% sodium chloride and 50% water. Here, 75% is sodium chloride and 25% is water. So children, now let's see the changes happening in each beaker. In the first beaker, how the water will be moved, where it is inside or outside. Let us see. See, here inside the cell having 80% water and outside only 50% water. Already I told you osmosis means the movement of water from the higher concentration to the lower concentration. That means water will be moved from the cell to the outside. So what will happen for the cell? What will be the result? The cell will start to shrink. The cell will start to shrink. So here let us see. Here inside the cell 50% water and outside the cell also 50% water. So the movement of water will be equal to outside and to inside. It will be equal. So there is no change for the shape of the cell. Now let's see here. Here inside the cell having only 25% of water, but outside it is 50% water. That means water will be moved from outside to the inside of the cell. Water will be moved inside the cell. That means the cell will start to swell. So the size of the cell will be increase. So children, see here the solution having 50% sodium chloride. Comparing to the cell, the salt will be more. So this kind of solution we can call hypertonic solution. Hypertonic. That means the solution contain more salt than the water. That is hypertonic solution. Now you can understand the cell kept in the hypertonic. What will happen? The cell will start to shrink. Now let's hear. If the salt concentration is equal to the cell, this kind of solution we can say isotonic. Iso means similar. It is equal. But here say the salt concentration is less in the solution, high inside the cell. So the salt concentration is less. That type of solution we can say hypotonic solution. That means hypo means less. Hypo means less. Hyper means more. So the cell kept in the hypertonic solution will shrink. If the cell kept in isotonic solution, nothing will happen. There is no change for the shape of the cell. If the cell is kept in hypotonic solution, the cell will start to swell. The size will be increased. If it is an animal cell kept in hypotonic solution, it will be burst out because there is no cell wall for the animal cell. But if it is a plant cell having cell wall, so there is no breakage of or there is no rupture of the cell. It will be just swell. Clear? Now let's do an experiment observing osmosis using an egg cell. Inside the shell of the egg, you can see a membrane. That is the plasma membrane of the egg. Now let's expose the semi-permeable inner membrane of the egg. We taken 2 eggs and kept it in a beaker. You know, the shell of the egg is made up of calcium carbonate. Now we are going to add acetic acid to this beaker. After the reaction of calcium carbonate and acetic acid, carbon dioxide, water and calcium acetate will form. It will take 3 days. So now the shell has only one outer covering, that is known as plasma membrane. Now we are going to placing these eggs in a hypotonic and a hypertonic solution. Before doing the experiment, let us take the weight of each egg. The first egg has 94 and the second one have 102. That is the weight of these two eggs. Then we kept the first egg in hypotonic solution and the second one in hypertonic solution. That means in the first solution it is only the water. Second one it is a corn syrup. We have to observe this experiment for 8 hours. Now we are going to take the weight of each egg. So the first egg we kept in hypotonic solution. Its original mass was 94 gram, but now it is 97 gram. That means the water will enter the cell. Now the egg kept in hypertonic solution. Now its mass is 59 gram, but its original mass was 103 gram. That means 57% change was there. So when a cell is in a hypertonic solution, water will leave the cell and the cell will shrink. Its size will decrease. Children, today we have learned how the carbon dioxide and oxygen and water movement inside and outside the cell, and what is diffusion, what is osmosis, and what is three kinds of solution: hypertonic solution, isotonic solution and hypotonic solution. So children, learn well. Have a nice day. Thank you.